So in the last video, we discussed the different reasons why cells reproduce, as well as the majority of where a cell spends its lifetime, which is during interphase, where it's either growing and then the DNA is doubling and it's preparing for mitosis. So all of these things need to happen before the cell can duplicate. So without further ado, let's get this mitosis party started. So there are actually 4 phases that are included in mitosis, and they go in this order. And if you remember the acronym PMAT, you'll be able to easily recall the order in which they happen. So P stands for prophase, M stands for metaphase, A stands for anaphase, and T stands for telophase. So if you remember PMAT, that is the specific order that mitosis happens in order for the cell to accordingly duplicate. So without further ado, let's get started. So for prophase, this is the very first phase. Now, if you look over here at my diagram on the right, you will notice that there are several descriptions over here. And one thing I do want to point out in terms of this is that this is now our nucleus right here. So this is the nucleus, and this is our mitochondria. These are our RER and SER, but everything has already doubled. The DNA has doubled. It's preparing for mitosis to begin. This is our very first phase in this. So one thing I do want you to notice is I want you to see these like squiggly like X things, okay? So this is going to be our condensed DNA, which we are going to call chromosomes. So the chromosomes are the condensed DNA, and they are making this X-like shaped thing because these are the doubled strands of DNA that happened during synthesis, the synthesis phase of interphase. So those are our chromosomes. And the one other thing that I want to point out here is that this organelle, remember we talked about those microtubules, okay? So these microtubules over here are their specific type of microtubules, and these are called centrosomes. Now within the centrosomes, there's going to be these specific little organelles called the centrioles. So the centrioles are going to create these spindle fibers that are located within the centrosomes. So I know there's a lot of C words here, so we'll have to try and keep them all organized. But that is kind of what's happening in the diagram, just so that you can kind of understand what is going on. So in prophase, the very first thing that happens is those doubled DNAs that have condensed now into chromosomes are going to meet at the centromere. And here's another C word, sorry, but the centromere is like this, it's like you can't really see it in here, but it's this like little connection point where the 2 X's are. That's the centromere. So the condensed chromosomes have now met there, and they are connected at the centromere. Now that they're there, the nuclear membrane is actually going to begin to dissolve, because if you think about it, if the cell's going to split, the nucleus can't stay intact. So that's why you have these green dashed lines right here, is to indicate that the nucleus is dissolving. So you can see that there. And then the other thing that happens in prophase is those spindles start to form from the centrioles. So these spindles are going to be really important. These are going to extend from the centrioles, which are located within the centrosomes, and they are going to continue to make more and more spindle fibers. Kind of think of it like Spider-Man releasing its web. Okay, so this is prophase. Now, after prophase, the cell still needs to do some preparations before it can actually split into 2 new cells. So the next phase that we have is metaphase. Now, M for metaphase, I always think of middle. Notice how things are lined up right here in the middle, right? Okay, so when you think of metaphase, think middle. That will help you remember what is going to be happening during metaphase. So in metaphase, the nuclear membrane is completely gone. So remember, it started to dissolve in prophase, and then so in between prophase and metaphase, the nuclear envelope has completely disappeared. The chromosomes, remember, which is that condensed DNA, line up in the middle of the cell. So that's why it's called metaphase. And then from there, you'll notice that a couple of things, hopefully, that first of all, our centrosomes that have those centrioles located inside of them have now moved to opposite ends of the cell. And these are the spindle fibers now that they are continuing to extend. You can notice that they are much longer than they were in prophase, and they're going to continue to extend and extend and extend and extend. Okay. Remember once again that our chromosomes are connected at their centromere. You can see it better. That's kind of like outlined in yellow in this diagram. So the chromosomes are lined up in the middle, and the spindle fibers from those centrioles continue to extend. They're closer and closer and closer to where the chromosomes are lined up in the middle. So that's what happens in metaphase. So metaphase, middle. The next phase is anaphase. When you think of anaphase, I want you to think A, away. So anaphase, A, away. With this, what's happening here is that the chromosomes now are splitting. Remember, we're trying to make two identical cells. So the DNA that has doubled is now going to pull to opposite ends of each of the cells. So the spindle fibers actually attach at these centromeres right here, and they're kind of like a hook. If you think about it, like when you would fish, the hook has now attached to that centromere, and it is pulling those chromosomes in two opposite ends of the cell. And those centriole spindle fibers, like I mentioned, are what are doing the pulling. Notice also too in this diagram here, it's called a cleavage furrow. Remember, in the metaphase, it was still one big circle. Now this is all the cytoplasm floating around in here. The cell is now going to start to cleave, or where it's going to start to indent in, and this is called a cleavage furrow. So what is happening is that the sister chromatids, which are each of the sides of the chromosomes that are attached at the centromeres, are being pulled apart by the centrioles, spindle fibers. So now that they've pulled apart, this is called a chromatid. Alrighty. So there were two of them when they were connected. That's why they were called sister chromatids. And now they are going to be pulled apart. So you're going to have chromatids on each side of your cell working its way there. And the last phase of mitosis is telophase. And during telophase, here's what's happening. There are two new cells that are beginning to develop, but they still share cytoplasm. So telophase, start to think of like the start of two new cells. The nuclear membrane's also starting to come back. You can see that with this dashed line around here. That's the nuclear membrane that's starting to come back. So you can see that there. And what's happening is that the nucleus is starting to reform. Notice now how there are similar things that are on each side, and the spindle fibers are starting to retract. And finally, now mitosis is over, and we call this cytokinesis. So in cytokinesis, remember, we have that mother cell that is now going to make two new daughter cells. So the cytoplasm has completely separated itself. The nuclear membrane is complete, and there are two new cells. The cells no longer share the cytoplasm, and they are identical to each other. Remember, because we're trying to make exact copies of one another. Take a minute if you're at home to take a look and see how this may look. These are actual slide images of these mitosis onion root tips. You can see the different phases of mitosis actually happening within the onion root. And then try yourself to see if you can identify the variety of these different phases of mitosis. Good luck.